Hello everyone, my name is Tatiana. I am a PhD student at the University of Aberdeen and I'm supervised by Professor Jenna Gregory. And today I want to talk to you a little bit about our research into MND and about how we look for clues in the body to find answers for the brain. I will be predominantly talking about the gut or the gastrointestinal tract, as that's my main research focus, but I will touch up on other organs as well. So to start off, what do we know about MND? Thanks to extensive research and clinical efforts, we know quite a lot these days. And here on the screen, I just put some basic information about MND. So we know that it's associated with movement problems. We know that it's the neurons in the brain and the spinal cord that are affected. We know that each patient has a completely different experience with MND. And we know quite a lot about the genetic and pathological factors that contribute to the disease. And here I highlighted TDP43 pathology because it's something that I'll keep coming back to again and again in this presentation. And just to briefly explain what it is, so TDP43 is a protein in the body that in MND changes its structure and it forms these toxic clumps that we also call aggregates. And because they're toxic, then they are then involved in the cell death or the death of neurons. And TDP43 pathology is present in 97% of MND cases, so it's very, very common. Unfortunately, another thing we do know about MND, as well as other neurodegenerative diseases such as Parkinson's or dementia, is that a lot of neurons are lost by the time symptoms appear. So this really means that if we want to intervene, if we want to find treatment for these diseases, we have to intervene as early as possible. But it's very, very hard to intervene in patients that don't have any symptoms yet. So ideally, really what we would want is a time machine to go back, 10 years before these patients got their symptoms and see what MND looks like at this point. So unfortunately, in our lab, we don't have a magical time machine, but we have something else in Scotland that we can use. So I think a lot of people have had a tissue biopsy taken before. It's a very, very common procedure. So in case you had maybe a suspicious looking birthmark, the doctor ordered for it to be taken and assessed diagnostically. So it's quite a lot of organs that we can take a biopsy from, but importantly, not the brain or the spinal cord. When a piece of tissue is taken for a biopsy, it's of course assessed diagnostically. But access tissue is actually stored in biobanks and biorepositories. And under the Scottish legislation around human samples, scientists are able to get access to some of this tissue and use it in research. And it is exactly what we do in the Gregory lab. So here I'm showing you a publication where we've taken archival biopsy tissue from MND patients, but these biopsies were taken 1 to 14 years before MND patients got their diagnosis. So you see how we're really trying to travel back in time in this case. And we have assessed this tissue for signs of TDP43 pathology. We found that skin, lymph node, gallbladder, and colon tissue all showed signs of TDP43 pathology, again, years before diagnosis. We have recently repeated this experiment now with an RNA aptamer instead of an antibody, which is basically just a more sensitive detection method. And we now found more tissue types that show signs of this early TDP43 pathology. Here I just want to quickly talk about the skin. So this is not my research, it's actually led by Christina and Katie in our lab. And in this original publication I showed you here, we used, I believe it was 8 skin samples from 7 different MND patients, but they now actually expanded the cohort to 25 samples from 17 individuals. And here in brown, you can actually see the TDP43 pathology, and this is in the sweat gland. And this is really exciting because Christina and Katie are now working on developing this into a potential early diagnostic test. But I'll jump back to the colon now. This on the right is a figure from this first publication that I showed you. So on the top here, we see an H&E stain of colon tissue. And an H&E stain basically doesn't detect anything in particular, any particular protein or anything like that. It just shows you the rough structure of the cells in the tissue. And this is also what a pathologist would see when they're shown a biopsy sample. So if you have a biopsy, it goes to the pathologist, and this is what the pathologist would see. And if you were a pathologist, you would be able to tell that there are absolutely no anomalies or pathologies visible in this picture. But now here on the bottom, we see the same tissue, the same sample, now stained with TDP43 antibody, so a detection method for TDP43 pathology. And now these brown clumps again appear. So there is TDP43 pathology. But of course, in this context, TDP43 antibody is not used in the clinic. So is it that we are completely missing this hidden TDP43 pathology years before these patients develop motor symptoms? As I mentioned before, my research is very much focused on the gastrointestinal tract, so I just want to introduce it a little bit more and tell you why it's relevant in MND. So certain MND patients experience digestive problems before their MND diagnosis. As I just showed you, GI tract tissue shows evidence of TDP43 pathology, again, even before MND symptoms. And much like the rest of the body, apart from the brain and the spinal cord, the gut is available for a biopsy. And actually, intestinal colon biopsies are very, very common in the clinic. It's a very routine procedure. And that really allows us to see what's happening inside the tissue in living patients. Because of course, when we talk about brain or the spinal cord, we can't get that tissue until the patients have passed. But now we really have the unique opportunity to look at tissue from live patients. And lastly, I just want to introduce a paper that associated digestive issues with a heightened MND risk. So it's this first publication here. It's a Swedish population study that identified a group of patients, a group of people that had digestive symptoms severe enough that they had to have a colon or an intestinal biopsy, but this biopsy was determined to be histologically normal. Well, these group of individuals had an increased risk of developing MND later on in their lives. And if I just take you back to the figure I showed you in the previous slide, if you think back to it, could it be that these group of individuals, even though nothing was noted on their biopsy, they actually had this hidden TDP43 pathology in their intestines the entire time, but it went completely unnoticed? So taking all that into consideration, this is my research, and this is how I leverage all the information I just showed you. So we built a new cohort that's very similar to the one I just described. So it's people aged between 60 and 70 years that have had a colon biopsy that was histologically determined to be completely normal. So no pathologies observable on the biopsy. We now have samples from just under 200 individuals in this cohort. We took their colon biopsy tissue, requested it from in the NHS Grampian biorepository, and we treated this biopsy tissue for signs of different protein pathologies. So the first protein we looked at was TDP43, as I mentioned before, associated with MND and dementia. But we also looked for signs of alpha-synuclein pathology, alpha-synuclein being mainly associated with Parkinson's disease, and tau pathology as well, which is mainly associated with dementia. And these biopsies that we investigated were actually taken roughly 15 years ago, which allowed us for some clinical follow-up time. And we asked a group of surgeons from the NHS Grampian to get us some clinical data on these patients. And so they got us more information about these people's biopsies, about their neurological health, as well as their survival. And here I want to start showing you the results that we found. So here we can see pictures of the pathology that we looked at. Here again in brown, these brown clumps are the TDP43 pathology, and here on the bottom, the brown is alpha-synuclein pathology. Alpha-synuclein was actually the most commonly pathologically present protein in this tissue. It was present in 46% of the samples. And TDP43 pathology was present in 34% of the samples, so roughly a third. and tau was not very prevalent at all. But taken together, 60% of the people in this cohort had at least one type of protein aggregating in their gastrointestinal tissue. And then 20% had 2 or 3 of these proteins aggregating simultaneously in their gut. And this is really important because these people, after they had their histologically normal biopsies, they were told that there's nothing wrong with them and they were likely discharged from the clinic. But in the meantime, they had this protein pathology that's associated with neurodegenerative diseases in their gastrointestinal tract. But how does this data correlate with the clinical data we have? So in the general population, we would expect 13% of people to get a neurological disorder. And when I say general population, I mean the same age as our cohort, so between 60 and 70 years. But in our cohort, 38% of the people were referred to neurology with symptoms of neurodegenerative disease. And among the diagnoses that we actually recorded, the most common ones were non-Alzheimer's dementia, mood disorders, or vascular dementia. Now, I want to point out that you can't specifically see MND on this list, but in the non-Alzheimer's dementia group, there were a couple of cases of frontotemporal dementia, which is a disease very much associated with MND. And there were also a few cases that had unexplained speech impairments or unexplained motor issues that just weren't diagnosed yet. So what I've shown you till now was that in this new cohort that we established, there is evidence of protein pathology in the gut. I showed you that the same cohort also has an increased incidence of neurological diseases. But lastly, the question remains, can the presence of protein pathology in the gut predict the onset of these neurological diseases? So here, this is a bit of a technical slide, but all you really have to focus on is this red dotted line here. And in case of alpha-synucleinopathies, which would be Parkinson's disease or Lewy body dementia, and then non-Alzheimer's dementia, which again, this group contains cases of frontotemporal dementia. For these two conditions, the bar is touching or above the red line. So this means that these two groups of disorders we can predict with the gut protein pathology. So if a patient had the gut protein pathology, they will go on to get some of these diseases here in these two groups. And lastly, here is really the most important piece of data that I want to share with you today. So this is a survival curve, and it shows the survival of a particular group of patients. So this here is the number of days, so the timeline, and you sort of see how many people are alive at what point. Now, if the next curves I'm going to show you are above this black line, that means that this new group of patients lives for a longer amount of time. But if the new curves are below, that means that they die faster. And this black line represents the people in our cohort that had no protein pathology in the gut. So here are survival curves for people that had only one type of protein aggregating in the gut, either TDP43 in red or alpha-synuclein in blue. And then here are the curves for people that had multiple proteins aggregating in the gut. So in purple, we see the survival of people that had 2 proteins aggregating in the gut. And then in green, these are people that had all 3 proteins aggregating in the gut. So TDP43, alpha-synuclein, and tau being pathological at the same time. So this is really quite a staggering survival difference. And what I wonder here is if we took these patients back when they had their biopsy, if we immediately were able to detect that they had this protein pathology in the gut, and if we introduced an intervention at this point, but it doesn't have to be anything MND specific, even if it's something very safe, for example, an increased fiber intake, would we be able to prevent the onset of neurological diseases in this group? Or would we be able to prolong these people's survival? Because, irrespective of neurological diseases or irrespective of MND, these people are dying much quicker than the controls in this cohort. So here I just want to finish up a little bit with saying how we can use these findings. So we can use this gut protein pathology to predict certain neurodegenerative diseases, like I showed you. We can also check, we can also use this in clinical trials to check if the new treatment that's being tested is working in clearing the proteins. And we can also use this as general health marker. So again, these people, irrespective of neurodegeneration, they are dying very fast. So I just want to conclude this presentation here. So what I talked about first was how in our lab we go back in time with biopsy tissue, and I introduced you a little bit to the skin research that's going on in the group as well. Then I introduced you to this novel cohort that consists of people that had a colon biopsy, but this biopsy was histologically determined to be normal. And I told you that there is evidence of protein pathology in these gut samples. I also then told you that there's also evidence of increased rate of neurological diseases in the same cohort. And lastly, I showed you that we can use this protein pathology in the gut to predict certain neurological diseases and shorten survival as well. And with that, I just want to thank you for your attention. I want to thank all the members of our team that are, again, supervised by Professor Jenna Gregory. I want to thank all of our funders and collaborators, and of course, all the people donated their samples, because without them, this research wouldn't be possible. Thank you very much.